Welcome everyone. All over the world, plastic pollution continues to build up in oceans, landfills, and even within our own food supply. Traditional recycling methods cannot keep pace with the billions of tons of plastic waste being created every year. Now scientists are turning to a radical solution based on living organisms. Certain bacteria have been discovered that can actually consume plastic and convert it into harmless byproducts. These microbes have enzymes that can break down the molecular structure of polyethylene terephthalate and other common plastics. If properly engineered and deployed, they could dramatically reduce the long-term threat posed by synthetic waste without relying on costly industrial processing. The discovery of plastic-eating bacteria originated in a Japanese waste facility where researchers identified a strain of Ideonella sakiensis capable of breaking down polyethylene terephthalate. This type of plastic is used in bottles, food containers, and synthetic fibers. The bacteria produce two enzymes which act in sequence to depolymerize the plastic and then metabolize the resulting compounds. Once this process was observed in a natural setting, scientists realized the enormous potential for using similar organisms on a large scale. The goal became to enhance the efficiency and durability of these strains through laboratory research. To make bacteria-based plastic digestion viable for industrial use, researchers began modifying the genetic code of these microbes. Using tools like CRISPR and protein modeling, they increased the rate at which the enzymes could digest plastic under varying conditions. One focus was enabling the enzymes to function effectively in lower temperatures and fluctuating pH environments. This adaptability would allow the bacteria to survive and work in more realistic settings such as municipal landfills, riverbanks, or marine waste zones. Successful strains could be produced in bioreactors and then released into controlled waste treatment systems. Deployment in landfills is one of the most promising applications. Rather than simply burying plastics where they take centuries to degrade, engineered bacteria could be introduced in moisture-controlled zones. These environments would be maintained with nutrient substrates to keep the microbial colonies alive and productive. As plastic waste breaks down, the bacteria convert it into simpler organic molecules such as carbon dioxide, water, and biomass. Unlike chemical recycling, this method does not release toxins or require intense energy input. It transforms a passive waste pile into an active processing environment. In aquatic environments, the situation is more complex due to dilution and mobility of water. However, researchers are developing methods for binding bacteria to biodegradable rafts or slow-releasing capsules. These platforms can be placed in areas with high concentrations of plastic, such as ocean gyres or river mouths. The bacteria slowly emerge, colonize floating debris, and begin enzymatic digestion of plastics. This method avoids uncontrolled spread of genetically modified organisms and allows monitoring of microbial activity. The rafts can also be collected once their work is done, minimizing ecological disturbance. Factories that produce plastic waste during manufacturing could adopt on-site bacterial digestion as part of their waste management strategy. By integrating custom tanks filled with optimized microbial colonies, production facilities can directly degrade offcuts, packaging waste, and defective products without external processing. This closed-loop system reduces transportation costs and emissions, while allowing near real-time conversion of waste into harmless byproducts. Factory managers could monitor performance digitally, adjusting conditions such as temperature and nutrient availability to optimize the breakdown rate. In time, this could become a standard feature of responsible industrial design. Municipal recycling centers often struggle to deal with plastics that are contaminated, multi-layered, or too degraded for traditional recycling. Bacteria offer an alternative by targeting the chemical structure of plastics rather than relying on sorting and melting. Specialized processing zones could be set up where unsortable plastics are shredded and then exposed to bacterial enzymes. This would allow cities to extract value from waste that would otherwise go to incinerators or landfills. The resulting biomass could be composted or used in bioenergy production, completing a beneficial cycle for the environment. The enzymes themselves could also be used outside of living bacteria. Some labs are isolating the key proteins responsible for digestion and applying them in purified form. These enzymes could be sprayed onto plastic waste in industrial settings or even integrated into washing systems for cleaning recyclable materials. This approach removes the need for live organisms and makes the process more controllable. It also allows the enzymes to be stored, transported, and used on demand. The scalability of this method is one of the reasons companies are investing heavily in enzyme-based recycling technologies. There are still challenges to address before global deployment is possible. For example, many plastics such as polyvinyl chloride and polypropylene are more chemically resistant than polyethylene terephthalate. Research is underway to identify or create new strains of bacteria capable of digesting these tougher materials. Another concern is the safe containment and termination of engineered bacteria once their task is complete. Scientists are incorporating genetic kill switches that deactivate the organisms after a certain time or under specific environmental cues. These safety mechanisms will be critical to regulatory approval and public acceptance. Environmental groups and governmental agencies are also being involved to ensure ethical and transparent use of the technology. Guidelines are being developed to prevent misuse or unintended ecological effects. In some cases, public testing areas will be monitored by academic institutions to provide real-time data and encourage public trust. Public education campaigns will likely be needed to help people understand how these microbes work and why they are safe. This effort will help reduce fear and build enthusiasm for a cleaner and more sustainable solution to plastic pollution. The long-term vision for bacteria-based plastic digestion involves a global network of waste management systems working in harmony with natural processes. Instead of fighting the mountain of synthetic waste through energy-intensive solutions, societies could embrace a biological method rooted in nature's own adaptability. Whether through live organisms, extracted enzymes, or hybrid systems, the result would be a world where plastic waste no longer lingers for centuries. Future generations may look back on this breakthrough as a turning point in how humans manage their materials and responsibilities to the environment. If this technology continues to evolve, it could lead to unexpected applications beyond waste disposal. For instance, imagine clothing that can decompose itself when exposed to a specific enzyme mist, or packaging that breaks down in compost bins without manual sorting. Even spacecraft or remote outposts could carry bacteria-based kits to manage plastic waste without needing resupply missions. In extreme scenarios, autonomous drones might be used to release enzyme capsules into illegal dumping zones or floating garbage patches. These ideas may seem futuristic, but they highlight the enormous potential of biologically driven waste control systems. Looking ahead, bacteria-based plastic digestion could become one of the most effective and widely accepted waste mitigation strategies on the planet. Instead of burying plastics or attempting to melt them down, future systems could spray or embed these microbes in landfills, oceans, and even factory waste streams. Advances in genetic engineering and environmental modeling will help ensure that the bacteria perform only their intended role without upsetting other ecological balances. As more nations search for scalable answers to plastic pollution, this biological approach may offer one of the cleanest and most natural solutions available. Thank you very much for watching. Please like and subscribe.